ATP. Rather, it results in the release of CO2 with the utilization of ATP. It leads to a 25% loss of the fixed CO2. Significance of photorespiration. 1. Photorespiration helps in dissipation of energy where stomata get closed during daytime because of water stress. 2. Photorespiration protects the plant from photooxidative damage by dissipating excess of excitation energy. Photorespiration is a process that occurs in Calvin's cycle during plant metabolism. In this process, the key enzyme rubisco that is responsible for the fixing of carbon dioxide reacts with oxygen rather than carbon dioxide. It occurs because of the conditions in which carbon dioxide concentration falls down and rubisco does not have enough carbon dioxide to fix and it starts fixing oxygen. Under suitable conditions, C3 plants have sufficient water, the supply of carbon dioxide is abundant and in such conditions, photorespiration is not a problem. Mechanism of photorespiration. 1. Oxygen fixation. In photorespiration, oxygen, O2, is mistakenly bound by the enzyme rubisco, ribulose 1,5-bisphosphate carboxylase oxygenase, instead of carbon dioxide, CO2, to the IUBP, forming a 2-carbon compound called phosphoglycolate. 2. Conversion to glycolate. Phosphoglycolate is then converted into glycolate in the chloroplast. 3. Peroxisome reaction. Glycolate moves from the chloroplast to the peroxisome, where it undergoes a series of reactions that release carbon dioxide and produce glycerate. 4. Mitochondrial involvement. Glycerate moves from the peroxisome to the mitochondria, where it is converted into glycine. 5. Return to chloroplast. Glycine returns to the chloroplast, where it reacts with another molecule of glycine to form serine, releasing ammonia in the process. 6. Conversion to glycolate. Serine is then converted back into glycolate, which re-enters the peroxisome to continue the cycle. The correct sequence of organelles in which glycolate and glyoxylate are produced sequentially in photorespiration is: chloroplast, IUBP reacts with fixed oxygen to produce phosphoglycolate, which is converted to glycolate. Peroxisome, glycolate is transported to the peroxisome and converted to glyoxylate. Peroxisome, the glyoxylate is converted to glycine by glutamate. Glyoxylate aminotransferase. Photorespiration is influenced by high temperature as well as light intensity and accelerating the formation of glycolate and the flow through the photorespiratory pathway. Photorespiration causes a light-relying acceptance of O2 and discharge of CO2 and is related to the creation and metabolism of a minute particle named glycolate. Photosynthesis and photorespiration are two biological processes in flourishing plants that can function simultaneously beside each other, as photosynthesis gives off oxygen as its byproduct and photorespiration gives off carbon dioxide as its byproduct, and the said gases are the raw material for the said processes. When the carbon dioxide levels inside the leaf dip to about 50 ppm, rubisco begins combining oxygen with a UBP as an alternative to carbon dioxide. The final result of this is that, as an alternative to manufacturing 2 molecules of 3 carbon PGA units, merely 1 unit of PGA is fashioned with a noxious 2 carbon molecule termed phosphoglycol. To purge themselves of the phosphoglycolates, the plant takes some steps. Primarily, it instantly purges itself from the phosphate cluster, transforming those units into glycolic acid. After that, this glycolic acid is transferred to the peroxisome and then transformed into glycine. The conversion of glycine into serine takes place in the mitochondria of the plant cell. The serine produced after that is used to create other organic units. This causes a loss of carbon dioxide from the flora, as these reactions charge plants' energy.
Thanks for watching. Keep tuned with Biozing for more updates.